India mainly is an agricultural country, we know it, and approximately 33% of India's GDP comes from agriculture. And the agriculture, it employs 62% of India's population. So that is 62% of Indian people are involved in agriculture. Now, the major problem faced by India after independence was to produce enough food for the growing population. But it with only limited land that is fit for cultivation. So that India tried to increase yield per unit area from whatever that existing farm land available. So what they did in mid 90s, they developed and introduced several high yielding varieties of wheat and rice. So they developed all by employing various plant breeding techniques, and we have already studied in the previous sessions what actually is plant breeding, right? And that resulted in the development of high yielding varieties of wheat and rice. And introduction of these high yielding varieties resulted in increase in the food production. So that phase where there is an increase in the food production, we refer it as green revolution. Let's see some Indian hybrid crops that are high yielding varieties. And the first one we shall see about wheat. Right, under the wheat during 1960 to 2000, the production of wheat increased from 11 million tons to 75 million tons. So it's a huge increase in the production of wheat, and this all it became possible due to introduction of the semi-dwarf varieties of wheat in around 1963. And these varieties are actually developed by Norman E. Borlaug, and he developed these varieties at International Center for Wheat and Maize in Mexico. Some examples of these semi-dwarf varieties of wheat include the Kalyan Sona and Sonalika. And these, as I told, are semi-dwarf varieties of wheat, and these are actually high yielding and disease resistant, and because of this the production increased from 11 million tons to 75 million tons. Now coming to rice, in the same period during 1960 to 2000, the production of the rice increased from 35 million tons to 89.5 million tons. And this all became possible due to introduction of those semi-dwarf varieties, right? And some of those varieties we'll see here. So one, it's a IR8 is a rice variety that was introduced in India, and actually it was developed at IRRI, that stands for International Rice Research Institute, located in Philippines, right? And another one, the Tai Chung Native 1, and this is again another rice variety, and that was developed at Taiwan, and these varieties were actually introduced in India around 1966. Now later, better yielding varieties like this Jaya and Ratna, which are also the semi-dwarf varieties, and these varieties were developed in India. So with the introduction of these semi-dwarf varieties from other countries and even developed within the country has actually resulted in a very high increase of rice production in India. Now coming to sugarcane, we have a species called Saccharum barberry, and this Saccharum barberry actually grown originally in North India, but there is a problem with this Saccharum barberry. It has actually a poor sugar content, and yield is also not to that extent. And another species named Saccharum officinarum. This has actually thicker stems and very good, okay, yield. So that is higher sugar content, and yield okay, is also good. Now, but the problem with this Saccharum officinarum is that when they try to grow this okay, officinarum in North India, it did not grow well. Okay, so now what they did is they crossed Saccharum barberi with the Saccharum officinarum, and they obtained a hybrid variety. Now this hybrid variety solved the every problem. It actually has thicker stems, and it also has a high sugar content and yield. Now this hybrid variety is able to grow even in North India. Right, so with this they also have increased the production of sugar in India. Now coming to millets, usually it includes maize, jowar, bajra and so. Now these we also have introduced lot many hybrid varieties of these maize, jowar, bajra and so. Here I just give you the examples for maize, like Ganga, Deccan, etc. So these varieties actually they are resistant to water stress, means they can even withstand with a very low water availability, that is in drought conditions also they are capable of growing normally, and they are also resistant to diseases. So this like this the introduction of these high yielding varieties actually led to the green revolution in India. Now let's look at plant breeding for disease resistance. Means we are going to use the technique of plant breeding to develop the plants with the resistance to diseases. Now what actually is the meaning of resistance? The meaning of resistance here, the plant, the host plant should have that ability to prevent the pathogen from causing the disease. The pathogen is a organism that causes the disease. Right, now the ability of the host, so from where does the host plant gets this ability? So the ability depends on its genetic constitution. Right, now what are all those pathogens that cause diseases in the plants? They come from, okay, the group of fungi. Then so that is the fungal, there are fungal diseases, there are bacterial diseases caused by the bacteria, and there are diseases caused by viruses, which we call them viral pathogens. Now when the plant is affected by the diseases, then the yield will get, okay, very less. In fact, so what will be the loss of the yield? So sometimes it will be around 20 to 30% of the crop loss can take place, or sometimes the total crop loss, that is 100%, okay, loss can take place. So for this purpose, we need to develop new cultivars that are resistant to diseases. Now, if there is a crop and it actually gives a very good yield, but the problem is with its resistance to diseases. Now, even though if it gives okay, good yield, of course, without the diseases, then if it is affected by disease, then there will be a lot of loss. So even though the plant has the ability to give a good yield, but it does not have the ability to resist the disease, in that case, okay, also the farmer will be losing the crop. So for that reason, we need to develop new cultivars that has the ability to resist the diseases. If it is the case, then it will give a good yield, and that actually results in enhancement in the food production. Right, so if we are able to develop new cultivars with disease resistance, then we can even reduce our dependence on fungicides and bactericides. These are chemical substances which we use on the diseased plants. So if the plant is infected with some fungal diseases, then we use these fungicides, okay, bactericides if it is in the case of bacterial infections and so. And moreover, these chemicals are also not environmental friendly. In fact, they can cause environmental pollution too. So for that purpose, so if we are able to develop the new cultivars or the new hybrid varieties that are able to resist the diseases, okay, so then we will have two plus points. That is, there will be increase in the yield, and again it reduces our dependence on these chemical substances. As I told, resistant to diseases depends on the genetic constitution of the host plant. So resistance to disease depends on the genetic constitution of the host plant. Now, before breeding we have to know about the causative organism. We should know about the causative organism and also the mode of transmission. So I just mentioned, so here is a note that before breeding is carried out, so before we start the breeding program, we should know about the causative organism. Okay, so information on causative organism and also the mode of transmission, means how this organism transmits. Okay, maybe through water, air, or maybe some vector that carries this disease from one plant to another plant. These are all okay the modes of transmission of disease from one plant to another plant. We should know about how that particular pathogen is transmitting from one host plant to another host plant. So this is important. So before breeding is carried out, information on causative agent, that is the pathogen, okay, the one which causes the disease, and also we should know about the mode of transmission of that disease. Now here I have given some fungal diseases. Now those fungal diseases in the plants we commonly call rust. So rust is a plural word. Okay, so all those diseases caused by fungi in plants, we call them rust. So here I have given some examples of the fungal diseases in plants. The first one, the brown rust of wheat, and this is caused by a fungus called Puccinia recondita. Then the red rot of sugarcane, this is caused by Coletotrichum falcatum. And another one, the late blight of potato, this is caused by Phytophthora infestans. Right, so here I have given an example for the bacterial disease in the plants. Right, so this is only for general information. So black rot of crucifers, this is the causative organism, Xanthomonas campestris. So here actually the crucifers are vegetables that belong to the family Brassicaceae, like cauliflower, cabbage, broccoli, and so on, right? Okay, so now let's see some viral diseases. Okay, so here the tobacco mosaic virus, that is caused by The tobacco mosaic is a disease that is caused by tobacco mosaic virus, and the turnip mosaic is caused by turnip mosaic virus. So here I gave some examples of fungal diseases, bacterial and viral diseases, so that harm the host plants, or I mean the cultivated plants a lot, and that results in a great loss to the farmer. Now here let's look at the methods of breeding for disease resistance. So how we are going to develop the plants, I mean the cultivars, for disease resistance, and this is actually carried by in two ways. One, the conventional breeding techniques, or they may use even mutation breeding. So the conventional breeding techniques may include the techniques like a hybridization, same, so which we have discussed earlier, like a transfer of the pollen, okay, I mean emasculation and bagging and so whatever, so that comes under hybridization. Okay, so this conventional breeding technique, it includes hybridization, and another one, it also includes selection. So by seeing the characters of the plant, okay, and observing them, we will come to know which plant has the disease resistant characters. So just okay, by looking at that or by observing the plant with the disease resistance, we will be selecting that plant. So that process we call it selection, right? And hybridization, we already know it, okay. And this mutation breeding, I'll talk about, so how they are going to perform this mutation breeding. The steps for developing disease resistant plants is same as that of, say, same as of plant breeding. So earlier I discussed about the various plant breeding steps, like a collection of germplasm, evaluation, okay, and cross hybridization of the selected parents and whatever. So almost the same steps will be followed here also to develop the plants for disease resistance. So the steps for developing disease resistant plants is same as of that, okay, plant breeding steps. So the steps are screening germplasm for resistant characters. Okay, so I told you already, the steps are similar to the plant breeding steps which I have discussed earlier. And the second step includes a hybridization of the selected parents, and then going for the selection and evaluation of the hybrids. And lastly, we need to test, okay, in the research fields and then in the farmers field. Okay, and then after we come to know that the plant is stable and able to give the best performance, then only they will be released into the market as new cultivars. Here I have given some crops that are resistant to diseases, right? So here these are the varieties that actually we have developed by that plant breeding technique, and we have given them names. So these are the new cultivars who have the disease resistance capability. Okay, so in case of wheat, we developed a new variety whose name is Himagiri, and that Himagiri variety is resistant to diseases like leaf rust, okay, and stripe rust and hill burnt are the diseases which actually it can fight with, okay, the Himagiri variety, and that's a wheat. Now coming to the second one, Pusa swarming. So this is the brassica, and it's also called currant rye, and this variety or the cultivar is resistant to white rust. Now another hybrid variety or the cultivar that is resistant to disease, okay, comes from this cauliflower, and is called Pusa shubra and Pusa snowball K1. Now this variety is, that is Pusa shubra and Pusa snowball K1, is resistant to black rot and curl blight black rot diseases. Now, and another one, okay, the cowpea, from where we developed a Pusa komal cultivar, and this is resistant to bacterial blight, right? And from the chili we have Pusa Sadabahar, and this Pusa Sadabahar variety is able to resist chili mosaic virus, and it is also able to resist tobacco mosaic virus, and it is also able to resist the leaf curl disease, right? So this is the variety. So these are the main, okay, the cultivars which are very important. We should remember, okay, so the crop and the variety, and also the resistance to which disease. This table, okay, is very important in case of NEET, as well as, okay, in board examinations. Now let us see the mutation breeding. Right, so first let's look at the definition of the mutation breeding. It's a process, right, by which genetic variations are created through changes in the base sequences. So the base sequences will include those nitrogen bases, as we have not finished the chapter molecular biology, so where we will study in detail about the DNA structure. I hope you have a little idea, as you have studied in first year, so what actually are these base sequences. So these base sequences are very important, and they are particular for a particular gene. So any change in those base sequences can result in mutation. So that's what, right, so we call it the mutation breeding. So through changes in the base sequences within genes, resulting in the creation of a new character. So by changing the sequences of that particular gene, we can create a new character, right? And that new character which is not found in their parents. So that's what is a process by which genetic variations are created through changes in the base sequence within genes, resulting in the creation of a new character that is not found in their parents. Okay, now so we understood what actually is the mutation breeding. So there are several genes in the plants, right? And we are going to modify some of those genes. So how we are going to modify? By changing the base sequences. So I told you already that each gene has a particular sequence of base sequence. So if you alter that base sequence, then it can result in the formation of a new character. A point that not all alterations can result in a formation of a new character. So okay, we'll understand those things a little later. Now coming to this conventional breeding, okay, the breeding that process we are doing already, okay, the conventional breeding. So this conventional breeding will depend only on the availability. So the number of genes availability is limited. So right, availability of number of disease resistant genes that are already present in the crop varieties, or maybe in the wild varieties. So in the crop varieties or in the wild varieties, those genes that are actually responsible for this disease resistance may be very much limited, and we depend only on that limited number of genes in case of this conventional breeding. Now okay, it has become possible to induce mutations. So here I told that change in the base sequence is what I call mutation, and that mutations can happen naturally, but now we are going to induce that mutations artificially. So it is possible to induce mutations artificially, and you can induce that mutations artificially, okay, either by using chemicals. So you can use some kind of chemicals, and also you can use radiations. So the radiations, like you can use gamma rays, okay, or there are certain kind of chemicals like base analogs. So here I'll mention. So for this, by using chemicals, I'll give examples like base analogs, hydrazine, etc. are some kind of chemicals which actually help in changing the base sequences. Right, so once we induce the mutations artificially, either by chemicals or by radiations, it will be followed by selecting and using those plants that have the desirable character. So if you find the character is desirable, so means with the use of these chemicals or radiations, if it is able to get success in the formation of a new character, then that's what the plants with the desirable characters, and you will be using them as a source of breeding. So the remaining process goes as usual that we discussed previously. I mean here as a source in the breeding process. Here in mung bean, it's scientifically called Vigna radiata. So in this plant, we induced resistance to yellow mosaic virus and also to powdery mildew. This powdery mildew is a fungal disease. So to these diseases, resistance is being induced in this mung bean with the help of mutations. Okay, so those artificial mutations which I have told, either by chemicals or by gamma radiations, we have induced the resistance to the yellow mosaic virus and also to the powdery mildew. Here, several wild varieties of different cultivated species of plants, they have certain already resistant characters. But the problem with these some wild varieties and different cultivated plants, that they have a very low yield. Now, if we have a high yielding cultivated variety, right, and this one has no resistance genes, then it may not be able to give the product or the yield, a high yield. Now if we are able to introduce these resistance genes into this high yielding cultivated variety, then we will be able to get a very high yield. So here a wild species of bindi, so this is okra, that has a resistance to yellow mosaic virus, and now transferring this to the Abelmoschus esculentus, that's the scientific name of okay, bindi, okay, from the wild variety, and they produced right a new variety, and they have given a name called Parbani Kranti. So this Parbani Kranti is a yellow mosaic virus resistant variety.